Hello and welcome to Learning Made Easy, where we make complex subjects simple and easy to understand. In today's video, we're going to talk about one of the most important concepts in immunology: antigen epitopes, haptens, and carriers. Antigen. A substance that can be recognized by immunoglobulin on B cell and T cell receptor on T cell when complex with MHC are called antigen. Antigenicity or antigenic reactivity. Antigenicity refers to the capacity of viruses to bind to specific antibody molecules. Immunogen. An immunogen is any substance that generates B cell, humoral, and/or T cell, cellular, adaptive immune responses upon exposure to a host organism. Immunogenicity. It is the ability to mount an immune response on encounter of an antigen. Example: Haptens acts as antigen but not immunogen. Factors affecting immunogenicity. 1. Foreignness. Effective immunogen must be foreign. Example: bovine serum albumin is human antigen but not in cow. 2. Size. Molecular mass less than 10,000 and not immunogenic. Molecular mass between 1,000 and 6,000 may or may not be immunogenic. Molecular mass greater than 6,000 are generally immunogenic. Most active immunogens tend to have molecular mass of 100 kDa or more. Small substances have decreased immunogenicity. 3. Chemical complexity. Heterogeneous molecules have more complexity and hence more immunogenicity. The greater the degree of complexity of protein, the more vigorous will be immune response. Carbohydrates are immunogenic only if they have complex polysaccharide. Nucleic acids and lipid require conjugated protein carrier. 4. Susceptibility to antigen processing, presentation with MHC. 5. Characteristics of host biological system. A. Dosage. Insufficient dose of immunogen fails to activate enough lymphocytes. Number of doses also affects outcome of immune response generated. B. Route of administration. Intravenous into vein, intradermal into skin, subcutaneous beneath skin, elicit most strong immune response, intramuscular into muscle, oral gastrointestinal route. See genotype of recipient. Epitope, antigenic determinant. These are specific antibody binding site at antigen surface. Immune cells do not interact with entire antigen molecules, instead recognize discrete sites or macromolecules on its surface are called epitopes. Epitopes are immunologically active regions of an immunogen or antigen that binds to specific membrane receptors on lymphocytes or secreted. These are protein segment of above 5 to 6 amino acid. B cell and T cell recognizes different epitopes. B cell directly interact with the antigen through its surface-bound immunoglobulin, whereas T cell bind to processed antigen complex with MHC. Antibody bind to epitope with weak non-covalent interactions and AMP, hydrophobic interactions such as hydrogen bonding, ionic interaction, disulfide bond, van der Waals force. Antibody and epitope have complementary structure. Epitope should not be too large, it may disturb the binding force. Types of epitopes of protein antigen. 1. Linear epitope. These consist of continuous residues on protein sequence. It is an epitope, binding site on an antigen, that is recognized by antibodies by its linear sequence of amino acids, that is primary structure. 2. Conformational epitope. These consist of residues that are discontinuous in the protein sequence yet come within close proximity to form an antigenic surface on the protein's 3D structure. Conformational epitopes are less obvious and more patentable than linear epitopes. Significance of linear epitope. For instance, if a protein sample is boiled, treated with beta-mercaptoethanol, and run an SDS page for the Western blot, the proteins are essentially denatured and therefore cannot assume their natural three-dimensional conformations. Therefore, antibodies that recognize linear epitopes instead of conformational epitopes are chosen for immunodetection. In contrast, in immunohistochemistry where protein structure is preserved, antibodies that recognize conformational epitopes are preferred. Paratope. In immunology, a paratope, also known as an antigen binding site, is the part of an antibody which recognizes and binds to an antigen. It is a small region at the tip of the antibody's antigen binding fragment and contains parts of the antibody's heavy and light chains. Each paratope is made up of 6 CDRs, 3 from each of L and H chains, that extend from fold of antiparallel beta sheets. Each arm of Y-shaped antibody has identical paratope at end. Affinity and avidity are terms used to describe the strength of binding between an antigen and an antibody. Affinity refers to the strength of the interaction between a single antigenic epitope, a specific site on an antigen, and a single binding site on an antibody. It is a measure of the strength of the individual bonds between the antigen and antibody. A high affinity means that the bond is strong and will not easily break, while a low affinity means that the bond is weak and will easily break. Example: the affinity of a specific antibody for a specific antigen can be measured by determining the concentration of antibody required to bind to a fixed amount of antigen and reach a half maximal binding, kD. A lower kD value indicates a higher affinity. Avidity refers to the overall strength of the interaction between multiple antigenic epitopes on an antigen and multiple binding sites on an antibody. It is a measure of the combined strength of all the bonds between the antigen and antibody. Example: a virus particle has multiple copies of the same antigenic epitope. If an antibody has multiple binding sites for that epitope, it can bind to multiple copies of the antigenic epitope on the virus particle. This results in higher avidity, meaning that the overall strength of the interaction between virus particle and the antibody is stronger, and virus particle is more likely to be neutralized by antibody. Haptens. The term hapten, half antigen, was introduced in the 1920s by Karl Landsteiner to define low molecular weight chemical reagents which, as such, will not induce immune responses. These are minute molecule that elicits an immune response only when attached to a large carrier molecule. Example of haptens: dinitrophenol, aminobenzene, penicillin, etc. When haptens are combined or bonded with carrier protein, then it is capable to elicit immune response. Hapten plus carrier protein, example BSA, forms hapten carrier conjugate, a potent immunogen. Hapten carrier conjugate can cause production of antibodies specific to hapten, mostly formed, antibody specific to carrier and antibody specific to conjugate. Common carrier protein: bovine serum albumin, flagellin, hen egg white, lysozyme, sperm whale myoglobin, tetanus toxoid, ova albumin, etc. Difference between haptens and carriers. 1 definition: a hapten is a small molecule that is unable to stimulate an immune response on its own, while a carrier is a larger molecule that helps deliver the hapten to the immune system. Size: haptens are typically small proteins or organic molecules, while carriers are larger proteins, polysaccharides, or other large molecules. Function: the main function of a hapten is to stimulate an immune response when bound to a carrier, while the function of a carrier is to enhance the immunogenicity of the hapten. Stimulating immune response: haptens cannot stimulate an immune response on their own, but they can do so when bound to a carrier. While carriers enhance the immunogenicity of the hapten by increasing its size, adding stability, or providing a means of presentation to the immune system. Immunogenicity. The immunogenicity of a hapten is enhanced by the carrier, which acts as a delivery mechanism for the hapten to the immune system. We hope you found this video informative and learned something new. If you did, be sure to hit the like button and subscribe to our channel for more videos like this. Thanks for watching.